Bonjour à tous. Donc aujourd'hui, nous allons écouter Wei Jen CHEN, qui nous vient de l'Université de Taïwan. Il va parler de la biodiversité marine des poissons de l'Indo-West Pacific et d'autres organismes. Wei Jen, c'est à toi.
C'est là? Ah ouais, ok. Donc je parle français ou anglais?
Comme tu veux.
Ok. Ouais, je parle. Je mixe alors. Okay, so good morning, good afternoon, everybody. Wei Jen CHEN, and this is the title of my talk today: Marine Biodiversity Exploration, Conservation, and Evolution of Indo-West Pacific species and other organisms. En gros, c'est à peu près le travail que j'ai fait depuis derniers 10 ans, en fait, quand j'étais à Taïwan. Ok, donc je présentais un peu ce que je travaille ici. À Taiwan, pas ici en fait, à Taiwan. So this talk I will present some background about Indo-West Pacific Ocean, and I explain why we are interested in this area, and some research goal of my research, and short presentation about our international cooperation program, especially with the French team, tropical deep sea basin, and then some empirical study about marine biogeographic study, and some ongoing results that I developed since the last 5 years about the eDNA, environmental DNA approach, and some future outlook to conclude my talk today. So about the background, why we are interested in Indo-West Pacific Ocean. So because, as we know, Indo-West Pacific has the highest species diversity in the world. So the marine geographic province, which has the highest marine biodiversity, especially in the central part of IWP, actually we call Coral Triangle. The north extends to the Taiwan, and then the eastern part extends to New Caledonia, and the west part extends to Bali and Indonesia. So to show up, okay, the amazing species diversity in this region. Here we have at least 500 species of reef-building corals. So that's why we call it Coral Triangle. Okay, the highest coral diversity region in the world is about 40% of species in the total of the world here. And then we have about the fish we are interested in. There are about 37% of world's coral fish can be found in IWP, Indo-West Pacific region. 7 out of world's 7 marine turtle species. So the picture is I took from the Taiping Island, which is the southernmost territory of Taiwan in the South China Sea. So such kind of like species diversity and ecological diversity are quite important feature for us for the consideration in the basic research, such as the biodiversity, biogeography, and evolution biology of the marine organism. That's why I'm interested, and you guys are interested in this area. Okay. And on the other hand, IWP's rich diversity, however, or biological and non-biological one, are also widely used by people. It provides also important ecosystem service for human society, as you see from your labs. So including the fishery, tourists, and so on and so on. Okay, so for example, the coral reef ecosystem, we have estimate associated economic impact about this number, this US dollar amount per square kilometers of the reef per year. So according to this report, so coral reef ecosystem is a valuable ecosystem in the world, but is a vulnerable one because some human impact and so on and so on. So let's look at the human impact. When people use the ocean, they also cause serious impact on the marine ecosystem, such as pollution, habitat degradation, acidification, global warming, etc. But such kind of attention has not been fully paid by us, especially from the deep sea ecosystem. Okay. For example, this is the new quite recent article show that the deep ocean is warming slowly, but dramatic changes are ahead. Okay. let's look at some example about the pollutions in the Indo-West Pacific region and so on, or other habitat degradation. So those pictures were taken from northern Taiwan, the coral reef area, by our colleague, Vietnamese Dennis. So we see lots of the report like this. So for example, this like a fishing net tangle the coral and then cause the death of the coral. And also we also collect lots of like fishing gears from the coral reef in northern Taiwan. So according to a report by this guy in 2020, about 4.8 to 12.7 million tons of unmanaged plastic waste entered the ocean from the land, but the global ocean environment record only about 6.6 to 35.2 thousand tons of floating plastics waste. So what's going on with those like for those like plastic waste? According to a paper published recently by my colleague in NTU, Jia Yunke, they said the Pacific is the most serious region with litters with such kind of like floating plastics. Okay, more than 50% of debris continue to drag at the sea, which has a huge impact on the marine ecosystem. So in this paper, they do some simulation and found that okay, some plastic trash actually do not come back to the land. Okay, all those trash come from the land, but to the ocean, but do not come back to the land. And then most of them are circulate in the center of Pacific Ocean. Okay. And then of course, what we really learn about what we observe about the marine litter is only about 1% as we see from this picture. And where are the 99% of missing plastic? Okay, behind this ice rock. So some of the plastics were degraded by physical forces, by sun and something like this, and then were into the water, become microplastic, which was eaten by the fish, for example, or some of them will sink into the bottom and become parts of sediment on the seafloor. And then for those who did not like degrade it to a small pieces, they become mega trash, and then they will sit down on the floor. Okay, so to settle to the bottom of the sea, become a debris on the seafloor. According to this paper published by my colleague in 2012, we can identify some hotspots of distribution of litters. Okay, in the Gulf of Mexico, and then when we found where they come from, actually they come from the dock of the ship. Okay, so those parts are the target of the big ship. So the waste is not only from the land, but also from those ships who navigating on the sea. So let's come back to this example. Actually, in 2014, when we do the expedition in South China Sea, close to a remote area, a remote island in Taiping Island, we collected trash at a depth of about 1,100 meters depth. Well, at the remote island in the South China Sea, where we see so many trash, actually the weight, the mass of biological mass is less than the trash actually. So where did they come from? So according to another study, okay, say when they tracing the sources by taking the plastic waste that enter to the ocean due to the improper management in 2010, actually we can check each trash by their DNA, not DNA barcode, in their barcode, and we can find, okay, this trash come from which country, and so on and so on. And then we analyze the data, and we find out the number one contributors country is what is from China. Okay, and then the second one is Indonesia. Okay, and then how about Taiwan? we still don't know. We don't have enough data to document if we really contribute such kind of dirty signal now. Actually, when we look at, okay, this area is we call the coral triangle, but in fact, it's not a coral triangle. Again, it's called the trash triangle. And then we have rich diversity, but we also facing some problems that we met by the human. Okay. And then this is a survey we conduct in 2020. We collect the marine organism, benthic organism, as well as the marine trash. Yeah, because we right now, well, anyway, we don't have too many organisms, organisms to analyze. We have to analyze the trash, unfortunately. So we make a hotspot from the east part of Taiwan in the Taiwanese Strait, and then we find out that the western part, especially this locality, is kind of like hotspot for the marine trash, and so on. And then the variation of the hotspot depend on the season, depend on the current and other things. Anyway.
Is there any kind of degradation in the seafloor, the reduction of the size of the plastic different?
C'est-à-dire?
Did you understand?
No.
No, I was wondering if it was some kind of more degradation when when the debris are on the sea.
Degradation depends on if they can persist like this shape, then we can find from the seafloor. Otherwise, we will find in the water column as microplastics. Yeah, okay. So on the left, you will see different kind of the diversity of different kind of trash over there. And then when we analyze it, we count like a plastic account for about 99% of total amount. So plastic is a big issue, okay, in marine pollution. Okay, so here I give a brief summary, and I will try to show the research goal. The summary is, in the past decades, human disturbance has extremely altered our ocean community, that we rely on the food, transportation, and recreation. But such kind of attention has not been fully paid, as we just saw from my presentation, especially the deep sea ecosystem. Continuing our wise use of the sea depends on a better understanding of marine biodiversity, ecosystem functioning, and evolution of marine organisms, and the consequence of our instruction on the working marine ecosystem. So actually, it's our research goal. We want to have the data because the data currently is not sufficient to answer many questions, including the question about conservation. So we need the data, we need to analyze the data to get a full understanding about our marine ecosystem and also the impact by the human. And then we need some education, especially public education, to make up to the end of our goal is the policy. So for the regulation and so on, and even for design efficient marine protected area and so on and so on. So that's why we hope that our temporary result of the research can be used as an important reference for future work of government agency, the policy making. For example, the OCA, Ocean Conservation Administration in Taiwan, that I had a project with them, as well as for the purpose of conservation, rehabilitation, and environmental education in Taiwan, and other countries in the IWP. So the research goal, what I'm doing in the past 10 years is collecting the data, analyzing the data, and so on and so on. Try to, how to say, okay, for the future, we can propose some policy making for better protect our ocean. So, and then about the policy making and so on, we try to build a conversation between the policy making part and the scientific part. And then last week, I have been in CON, and Sarah too was in CON for this conference. Then we have researchers and some government agency sit down together to look at, okay, some like insight into our research and then conservation issue from in Taiwan and in France. So, okay, go to this slide. So how we collect the data, how we carry out our research. Okay, no money, we cannot make anything. So thanks to our funding agency from Taiwan, both right now it's called NSTC. It's our research funding agency in Taiwan, and OCA I just mentioned, government agency about ocean conservation administration in Taiwan. We can conduct our research work, okay? And then we also, and thanks to some international cooperation program, like a tropical deep sea pencil, and then another project I'm conducting with a French researcher from IRD is called phylogeography and systematic of emperor and large eye sea breams and other coral reef fishes. So I will briefly introduce these two programs and then show some empirical study or the result. Okay, another project I'm conducting is more like, well, and some political things, not really research, but yeah, 1,000-pound program, okay, to do some scientific education project with South China Sea and Southeast Asian people. And then since to this program, I can host international students, especially from Southeast Asia. And also, thanks to this program, then I have money to do a kind of long-term survey, and not money to do the eDNA around Taiwan that I'm going to present later in this talk. Okay. So that is my teamwork. Basically, for last two projects, I tightly collaborate with my colleague from IONTU, BNE Dennis. He's a French guy, but he's our faculty in the institute who working with coral reef ecosystem, ecology, and so on. And Zhiling Wei, like the tallest guy, who is the benthic ecologist, okay, who work on the marine debris and also the worms in the sediments. And myself and Jia Yinke, who working on the microplastic and big data analysis, for example, to monitor the marine debris floating in the ocean, and so on and so on. And then my colleague Chen Fenhuang, who is the physics, who is in physics oceanography, her specialist is acoustics. So we work with her to establish a station, observatory station. We can monitor the ecosystem by using acoustic signals, so soundscape actually. Okay, and my assistant Michelle Lin. Without her, I cannot do nothing. Okay, so research focus. Let's come back to the research focus. So these past years, I do a lot of biodiversity exploration and biogeography, with emphasis on marine species. Okay, from tropical IWP, from deep sea and shallow water. So from the shallow water part, this picture was taken when we do a survey in remote island around Madagascar, which we call the Tebas. Okay, and on the photo on the right side, we do the deep sea survey. My first deep sea survey with Sarah in New Caledonia. Okay, SCOD. Okay. So let's talk about the research on the deep sea part tropical deep sea benthol program. I'm currently in charge of theory research for this program and so on. So I'm not spend too much about this program. I think everybody know this. So I just want to mention that, okay, the Taiwanese partner joined this project in 2010. So at that time, I was not in Taiwan yet. So, but at that time, it's Professor Wei Chen and Dr. Wang Zhaoshao joined the research team and then work with French collaborators. And then and this program, I just want to show this. Okay, so we do the expedition to explain the tropical deep sea pencil program and then the historical expedition in the world from the challenging expedition, and then we do this expedition in Taiwan NTU. So the same expedition has been held in Paris in 2016 and then in Numea in 2019. So we do this series of expedition in Taiwan and hopefully in Kyoto University, maybe next year. Okay, so we make a plan. Okay, so this program was quite well developed until now, thanks to this research project. Okay, French-Taiwan collaboration project. Okay, with Sarah Samadi. And then I joined this program, tropical disease program, in 2011. And then since then, we conduct together all this expedition. Okay, so the color like this expedition organized by Taiwan's part, actually by myself. And then we continue to do this kind of like a oceanographic survey. And thanks to this program. So the research vessel we use, including the old NOR1, Ocean Researcher 1, managed by our university, and number 5, which was sunken in 2000, something like this anyway. Okay, Alice and then Atalon in 2019. So just want to show how we work on both. Okay, maybe you guys know what is going on both. So those picture was taken in our expedition in 2019, Sakizaya expedition around Taiwan. Okay, so that is what we collect from the deep sea floor, including some trash, of course. There are some trash with them there, and so on. Okay. And then there's some like image about the specimen. And then those are sampling sites that we conducted together since 2010 around Taiwan and South China Sea, and also in Papua New Guinea and in New Caledonia. Okay, so and then that's since 2021 we have our new boat. It's called New OR1. But c'est pas très original. Okay, New OR1 looks like this. So, well, anyway. And then, since this new research vessel, we have already surveyed this area, okay, in the northeast of Taiwan, and then a little bit in the South China Sea, in Formosa Bank, over there. Okay, Formosa Bank. Okay, and then I'm planning another cruise, which will be held this summer to survey this area. So it's called Colden Valley in the southern part. There's 3 small islands over there. And then the deep sea part down to 6,000 meters in the east part of Taiwan, and so on and so on. And come back here to to take a since trap in this area. We put the trap with sunken wood and the bones, organic and subject, to see if any interesting organism will probably inside or not. Anyway, so let's show a map about the tropical tissue pencil, all the sampling site we have that we have been unfed since this program to 2020, and so on and so on. Okay, so let's come back to the objective of our researches of this project, including the deep sea project and the shallow water project. So the first of all, we want to reduce the taxonomy impediment in the knowledge of deep sea biology, for example. Okay, and then of course the result show up will be the taxonomy discovery and some like new pattern of distribution of species. And the second, the objective of research is to show the exploration bias toward flagship environment, especially we are explaining later. That kind of like a bias will strongly distort our understanding, of course, of the pattern of biodiversity in the deep sea and other regions as well. The third one will be the search of original gradients of marine biodiversity. I will explain this later. Let's talk about the taxonomy part. So since these 10 years, we of course discovered some new species. For example, from the South China Sea, Marco Field Bank in the middle of South China Sea, for example, this guy. Okay, Kaido Burka stigma because it's called stigma because they are small white point. Okay, so the 4 species we described from us. So we published paper in 2019 in Frontier of Marine Science. And also more recently, we also described a deep sea eel species from the South China Sea in this locality. And then this slide, okay. And then in Papua New Guinea, we conduct 4 expeditions. And then, well, we found amazing high diversity of deep water flat fishes, okay, in the new Papua New Guinea. So the work is ongoing, and then we expect to describe 12 new species. Okay, so this slide was summary, kind of like what has been done before, and then the work has been done after my participation after 2011. So before 2011, okay, tropical DC pencil collect the specimen, and then about 125 papers published, and about 115 new species described by using or comparing the material collected from the this program. Okay, and this is named when after my participation, we collect the sample and organize 8 major sorting and taxonomy workshop. And then so far we have around 10,000 specimens collected, and then deposited in our museum, NTUM, National Taiwan University Museum, and so on. This is the people who participate in this workshop, including some Japanese colleagues. So that is what looks like the National Taiwan University Museum geological collection. We are moving to a new space, so we'll be more comfortable for host the research working in our museum. So that is summary. Right now, after 2020, all these like taxonomy networker have published more than 50 papers, okay, using the material we collected with 33 new species. Okay. Regarding our effort from my work, from my lab, we have described 8 new species. Okay, and of course there are many others waiting for formal description. For example, the 12 new flatfish species from Papua New Guinea. So this gives you some idea. So, well, after 2010, we have this research result. Okay, second of all, I want to share some empiric study about this subject. So in the past, the expedition, biodiversity expeditions were carried out, especially from some like a flagship environment, for example, Antarctica. Why? Because some political issue, or somewhere, for example, shallow water, coral reef. Because what? Because, well, coral reef fishes are more interesting to some people because they are beautiful. Okay, such kind of like bias, okay, will totally destroy our understanding for our biodiversity pattern. Okay, not only in the deep sea, but only in the shallow water, but also in the shallow water. So to show this kind of like effect, with my student Shindy, that I co-advised with Sarah Samadi in Nature History Museum here, we show this study and show this published paper. Incorporation of deep sea and the small size species provide new insight into the gastropod phylogeny, because before period to this study, the phylogeny of gastropod was established based on a big size specimen. Okay, well, and then we don't know, and then also from the shallow water species. So we don't know where it come from those lineage of gastropod species. So we included the deep sea mollusca species from this family, from this well, it's not family actually, from this group, Coquilinifonia, and we have some new result from this. So Coquilinia, well, it's comprised some basal gastropod. They primarily live on the sunken wood. So those are environment, deep sea environment that we pay less attention in the past. Okay, but now we have something interesting. So Coquelinidae contain 45 species in 7 genera. In IWP, there are 24 nominal species in 4 genera. But according to my another study, we found high diversity from this group. So that's the Coquelinidae looks like. They rely on the sunken wood and live in the deep sea. So by including those taxa for the phylogenetic analysis, that's the result come up. Okay, we found Coquelinifloria a sister group to another deep sea lineage, Neophorina. So this color means different habitat, cold ship or hydrothermal vent, and other like sunken wood associate species, biogenic substrate, deep water and shallow water in different color, and so on and so on. And then we found the Vestropoda, the ancestor of Vestropoda is from shallow water and then to the deep sea during the evolutionary history. And we also found that there are two times transition from the deep water habitat and from other ecological habitat to chemical synthetic habitat. Yeah, twice here and here, and so on, so on. Okay, so this study can be carried out with this like evolutionary history, interesting in evolutionary the deep sea evolution, thanks to the specimen come from the small size and the deep water lineage. Okay. Okay, so let's finish the deep sea part. Let's talk about the reef part. This project basically conducted with my colleague from the south, Montpellier, Philippe Bossat, is here. Okay, when he's still young. Okay, and then we conduct this study. So okay, those are the sampling site where I have been in the whole Indo-West Pacific Ocean, for example, in Batam, Papua New Guinea, in Taiping Island, in the South China Sea, and also in Oman, okay, and also the debates from here. And then thanks to this sampling effort, we can conduct the study. So the objective of this study is to, of course, the biodiversity exploration. And another one is the search of origin of IWP or coral triangle extreme concentration of marine biodiversity, and so on, to understand the unbalanced spatial pattern of marine biodiversity. So this figure shows, okay, the hotspot of marine diversity across different animal groups. So as we see here, the hot color means richest biodiverse species diversity in this region. So we see coral triangle actually one of most species richest area for the coastal fishes and the coral and so on and so on. So we want to know what coral triangle has the highest species diversity. We have couple hypotheses. Okay, for example, the first three are most dominant hypothesis to explain this diversity pattern. The first one is central or origin hypothesis. The second one is central accumulation hypothesis. The third one is provincial overlap hypothesis. So for the central origin hypothesis, we propose that the center of origin is the species diversification center. So as the center, then followed by outwarding migration to other region, to the peripheral region, to explain why there is so many diversity in the central part. For example, there's a continuity of speciation event happen in the central part, and then we can find more species in this area than peripheral area. The second hypothesis is to explain why here have such huge diversity in the central. we propose that speciation event happen in the peripheral area, then they have inward movement migration in the past to the central part. For example, if we have two species here, one species here, three species here, four species here, then each region, peripheral region, can contribute some species in the central part. Then we can see here we can observe more species diversity in the peripheral area. The third one is called co-functional overlapping hypothesis. In the past, okay, we have three biogeographic formats like this way, separated by marine biogeographic area. For example, in the middle of IWP, we have this geographic barriers called Sunda barrier that separate the Indian Ocean from the Pacific Ocean during the glaciation period. Okay, and then when the climate changes, sea level go up, there is a connection between Indian Ocean and Pacific Ocean. Okay, because of the sea level situation. So if we have, that means if we have 5 species in this geographic formats, 3 species here and 4 species here, then in the overlapping area, we can observe more species than other regions. Okay, to test this hypothesis, we need to know if the distribution, we need to know the historical biogeography to see if in the past there is a biogeographic barrier happen in this area or not. Okay. So the first hypothesis and the second hypothesis are alternative hypotheses. If we accept one, then we have to reject another. Okay, so it's called alternative hypothesis. But the third hypothesis is kind of like a supplementary hypothesis that can support either the first hypothesis and the second hypothesis. Okay, so normally we are testing the hypothesis 1 and hypothesis 2, and then reject one of them. Okay, so we conduct this study, diversity, phylogeny, and historical biogeography of large-eye sea breams with my colleague. And then this paper has been published in 2020. We sample the species from this group and from different localities in the IWP as I just showed previously, and then make this study. So I'm not going to give detail. So I just give you the conclusion. Conclusion is our results provide a new perspective on the evolution of this group, and with discovery of potential new species and new distribution records. For the biogeographic hypothesis testing, we reject this hypothesis, center of accumulation hypothesis, but we cannot reject this and then accept this hypothesis, central of origin hypothesis. Okay, because that historical biogeography analysis looks like this. So that is the time tree, and then this is the phylogeny, show the ancestral habitat of those current existing species. Okay, so like here, the color means, the green color means central IWP, the red color means Indian Ocean, and then the blue color means West Pacific. Once we construct ancestral habitat using the ancestral character reconstruction method, we can show this pattern. So we can find from here to here, the north central part, the central IWP are the ancestral habitat for those fishes. And then we conclude, actually, the central IWP is the central origin of this group of fishes. Then we have at least twice early stage expansion, one to Indian Ocean and then the other to the Pacific Ocean, and then come with couple of like a recent expansion from the central IW to the other two regions. So we confirm the central hypothesis. The diversity center is in the central because we found the ancestral habitat happened in the central of IWP. Then we have multiple outwarding migration events in the Estuarine history for this group of fishes. So that's why we accept this hypothesis. Okay, and then we also look at diversification patterns through the time, and we found out that after 70 million years ago, we had an acceleration of diversification rate to the present. Okay, so at the beginning of our species origin, well, the slope is like this. Then we have a sharp slope, means that there's a rapid diversification to the present. Okay, so we wonder if the monotacinae, the evolution of this group of fishes, is impacted by some past geographic event or climate change or not. We identify three major events, okay, to explain why there is an acceleration of species diversification after 70 million years ago. The first of the South China Sea expansion. So, since of South China Sea expansion, South China Sea before is not like what we see right now. So there's an expansion from 24 million years ago. So they push the South China Sea to what we know right now. And then they stop the expansion at this moment. And then at this moment, there are emergence of many South China Sea islands. Okay, before there's no islands, shallow water habitat in the South China Sea. It's the open ocean. And because of this movement, tectonic movement, they push the fiber, how to say, they push they have the driving forces for the formation for the reef islands in the South China Sea. So those islands are important to host the reef fishes. That's explain why there's such faster rate of diversification. Another one is mid-Miocene collision. Actually, there's another tectonic movement from the south, okay, and then push to the South China Sea. So these two forces, South China Sea opening and the tectonic movement from the south, will create those South China Sea reef island. And of course, once you have island, there are coral building, reef building corals coming up, then give the suitable habitat for the coral reef fishes for their evolution. So actually, we find this interesting result on our study. Of course, we also find some new species from our phylogenetic analysis, and then that's 2 weeks I was in Montpellier and described the new species with my colleague. So that's the result. We find a new species and describe it right now. It's called Ginocanus indicus. So by their genetic and by the morphology. So this is new species. This is his sister species, Cyprian species. Here we find from the morphometric analysis, there are 2 morpho groups clearly. And this is the new species distribution. And the sister species here has been described already, like this here. And then there's a second contact zone here in Aceh area. So also explain, well, once there's an effect of physical geographic barrier in the middle of Indo-West Pacific Ocean, after the glaciation, the sea become connected to each other. Then the species sister, they have the second contact area in the middle of South China Sea, in the middle of IWP for the 2 species. So that's the result also is explained support by third hypothesis, okay, provincial overlapping hypothesis to explain the high diversity in this agent because before there are two geographic problems, biogeographic problems, and right now they are overlapping. So, for example, here you have one species here, and in the middle, for example, here you have two species. Okay, so central origin hypothesis support the diversification pattern of this group of species, and then we have another supplemental hypothesis which is called provincial overlap hypothesis to support to explain why in the middle of IWP has highly species diversity for this marine species, marine fish group. Okay, I'm going to finish it. So my third part of my study is new tools to meet new challenges, emerging technology for managing marine ecosystem for resilience. Okay, so for the management of ecosystem, we have answered these three questions. What species or lineage are present or absent? Okay, it's about the work for the assessment of biodiversity. That's what I am presenting. Describe the new species, find the new records, and the new distribution of species for better management of ecosystem. And what we are doing right now. And also, we want to know their distribution across their geographic ranges. It's a biogeographic study. And also, right now, we incorporate some like marine parameters and with some discovery from the survey of marine pollution and then marine debris. So we want to answer what's the physical condition for organisms in this natural population. We have some on-site observation and other genomic study to answer this question, and so on and so on. And then the emerging tools we are using now different parts. For example, ROV, we can document in situ the environment underwater. Okay, and then we can also use the hydrophone to to analyze the soundscape in the water and to analyze the dynamic changes of the soundscape from a fixed stations, for example. And then we apply the eDNA and meta-barcoding analysis to better analyze the species diversity and so on and so on. So that is what we have from our institute for the instrument. And then that figure shows how we work for the eDNA part. Okay, and then we have also a diver collect the water for analyze the eDNA. Okay, so this part is from my ongoing project with Ocean Conservation Agency administration. And there's a video show our survey, but anyway, it's not working. It's okay. So, and then we also have a collaboration with MHN and default map for doing the seamount research, especially by employing the ROV. So that is a picture showing what we work to collect the eDNA as well as microplastics. So we collect the water sample, and then one part we analyze for the microplastics, and then another part of water sample we filter in, and then we collect the eDNA sample from the water. Okay, so that is the process. After filtering, we do the lab work, we do the DNA extraction, we using 12S as a marker to identify the species. We use NGS to do the sequencing, and then we use the MyFish platform to analyze the data. So the purpose is to do the exclusive investigation of fish fauna in Taiwan from this project, and then assessment of fish diversity at different spatial scale, which is alpha diversity, beta diversity, and gamma diversity. Okay, so to make sure our assessment using eDNA is correct, we publish paper navigating the scale of diversity in subtropical and coastal fish assemblage, invested by eDNA and visual survey. So we compare our result from eDNA survey and with the scuba diving, the image result, and then we look at traditional methods can perform well to reply the question about the alpha diversity, beta diversity, and gamma diversity, or is by the DNA, by eDNA, and so on and so on. So that is the conclusion we get. So eDNA exhibits an extraordinary power to explore the regional species pool, even sufficient replicate. Okay, and at the beta diversity level, all the methods, including the traditional method, okay, image survey underwater, all the methods successfully reveal the same spatial pattern of beta diversity and distant decay of the similarity in fish assemblage. Okay. However, in the alpha diversity level, actually, the species number in this locality in the sampling site, we should pay attention. None of method is capable of investigating the entire listings of fish fauna. So, for example, the challenge for eDNA is that sometimes we overestimate the fish diversity because the particular fish eDNA not exists in this area, but is from outside of this area, outside of this sampling site. But, well, DNA can survive, for example, 1 week and float to this site, sampling set, and then we capture it, and we got the result. It's kind of like a positive, false positive result. And also because the primer efficiency, sometimes we cannot detect the DNA from a particular species. Okay, because, well, primer did not match to the temperate DNA of this particular species, and then we cannot detect this species is false negative. Okay, because we know there's a species over there, but we cannot detect by DNA. Same situation happened to the traditional method because we observe this species just this moment, but they could present somewhere, for example, benthos cryptical species, the fish hiding under the rock, we never observe by visual survey, but we can detect it by eDNA. So that explains, well, alpha diversity, none of this method is capable for an entire survey for the resident fish fauna. Anyway, so let's come with our result. So our fish eDNA result have two parts for this project. Ship-based result, okay, and then coral reef result based on the diver sampling, based on the sampling by the diver. So since 2021, we have a total of 83 samples, okay, in 2 years, okay, around Taiwan with the research vessel, and then we have detected a total of 946 species of marine species, and surprisingly, we also detect 70 species of marine mammals, cetaceans. Okay, and let's look at the total record of fish diversity in Taiwan. It's like this. The total is 2,400 species. Then we reach about half of species diversity only by 2 years of the work. So that record is about maybe 30 years of survey by using traditional method, benthic trawling of fishing and other methods. Okay, and then this shows the saturation plateau of our of the species diversity, actually gamma diversity. So from the eastern part of Taiwan, western part of Taiwan, and the green is total. So only eastern part of Taiwan, we still haven't reached to the saturation point. That means we still have more investigation to do in the future for the eastern part of Taiwan because the water is quite deep and so on. Let's go back to the alpha diversity level that's around Taiwan, the site we surveyed in spring 2020, spring 2022, summer 2021, and summer 2022. So we can more or less know relative diversity among the cities in Taiwan. So Penghu here are diversity hotspot, and then northeast part of Taiwan hotspot of marine diversity. Okay, so that's the conclusion like this. We also do the beta diversity. So this NMDS illustrating, well, our ETN survey, the result do have geographic scales of like structure. Okay, eastern parts of Taiwan, western part of Taiwan, and the northern parts of Taiwan. Okay, so they show quite good resolution in beta diversity. And then for the reef survey, we have 72 sampling data from the important coral reef in Taiwan. And now we have 719 species detected. Okay, most of the common found species are from Labrador and the Formosan, which is what we expect, okay, from the coral reef area. So same for the saturation curve is like this here. Then that's fish ETN survey from the important coral reef in Taiwan. Again, Penghu and the southern Four Island area, well, host more fish species from our survey area. Same, we do the NMDS analysis and find very clear pattern in geography. Southern Penghu, northern Taiwan, Kenting and the Little River Island are clustered together, means there are some connectivity between these regions. Okay, and so on and so on. And then we also found that two sites are quite strange. TRM 20 and TRM 21 here, well, those two sites are very close to each other and located in east part of Taiwan. But one point is more like a group, more or less like with the northern part of Taiwan, which is subtropical area, coral reef area, and another one is more close to the tropical reef regions, points. So, 2 years is 21, that is the TRM 20 and 21. So 20 and 21 are in the north, are very close to each other, but in the NMDS, they are close to the northern and southern cluster respectively. This indicates that the fish assemblage in this area east of Taiwan cannot be determined solely by geographic distribution or temperate factor. Actually, that's the subtropical and the tropical part, the tropical region. So with another paper we published with Yanni and his student, we show that, okay, habitat change and its consequence on reef species specification in specialization in biogeographic transition zone. Actually, we show that, okay, tropicalization of benthic habitat, for example, stony coral and so on, does affect the specificity of the fish population. And under global warming, the distribution of some species, some specialists might shift in a manner that follow the movement of isothermals. That is, although the TM21 is located in the subtropical region, TM21 is located in the subtropical region, its benthic habitat may have been tropicalized due to global climate changes, and tropical specialized fishes has also been there. So to explain why TRM21 is located more close to the southern part than in the northern part. So tropical specialization species account for about 20% of the total from the ETN we detect from this region. So that means, well, global warming actually it happened right now in Taiwan's reef. Some small conclusion for the fish eDNA. Result fish diversity evaluated by eDNA show there are spatial and temporal difference in the presentation of diversity, yet spatial differences are more obvious than the temporal scale. So species diversity at an individual site evaluated by eDNA is consistent with the coral reef ecological survey result. Okay, we also survey the health of the coral reef. And then number of species detected from the site with the good environmental condition and or high protected intensity, for example, in the marine park in the southern Penghu area, is relatively high, indicating that eDNA could be an efficient tool for further monitoring of environmental stress and effectiveness of the MPA. So that is the short conclusion from the eDNA. And finally, for the emerging technology, ROV, we also have the survey with Gahin and Sarah in 2019. That is ROV we deployed in New Caledonia seamount, especially from these three seamount, Funida, Sea Duster, and the Monk D. So we don't have the analytical result yet, but just show the image. So that's what we found from the Monk D. Okay, with operational ROV. And then from Sea Duster, so we are lots of Sea Duster here, and we are ROV, and so on. And then it's from Munida. Okay, dominated by sponge, right? Okay, and so on. Anyway, to conclude my talk, so what I would like to say is new tools like ROV, eDNA, what is quite efficient to meet our challenges and then to manage our ecosystem. But those emerging technologies, okay, well, we can answer the question with precision, but more effort, for example, or long-term survey or monitoring are still required. Okay. Another challenge is, okay, not surprisingly, many new ways of doing science emerge. The difficult thing will happen. For example, so I listed three questions. No sufficient trend taxonomies exist for examining the correct organism data. For example, if we have DNA, eDNA data, we do not know exactly what kind of species there. We don't have a taxonomy background. Well, it's challenging as well. So anthropological impact is increasing. Okay, and so on and so on. And second one is the deciphering of our research has somehow been disconnected to each other. For example, well, there's some difficulty for the communication between the ecologist and the taxonomist, or something like we are the biologists, it's difficult to discuss with the social scientist, sometimes like this. Okay, anyway, so right now we are facing this rate of species extinction is much higher than rate of species discovery, which is true. So we have to continue our survey and so on and so on. And I think in a conceptual advance and the integrated approach are necessary to meet the new challenge, not only the new technology, but we need an advance in our knowledge and the integrated approach. So we are doing this kind of survey. I think if we don't do it now, we will regret it later, because in the future we have nothing to survey except the marine debris. Okay. And then, well, I will say that's political issue, but think more about a future Earth and ocean. So that is my survey. In next April, our research vessel is here. I'm touring in that area. Then they come up with Chinese military vessel and Taiwanese military vessel coming to see us. Okay, anyway. So right now, Taiwan is quite like, I think, hotspot for the political issue and so on. So I thank my lab member, okay, who contribute wonderful work for Kaido species description, Si Xuan, okay, and then my Malaysian postdoc, and then my Malaysian student who came back to Penang for her career already. Talk with my student from Thailand. Xin is my current PhD student from Taiwan, and Jin is from Hong Kong. So we are international team. And Xin is where is Xin? Okay, who contribute wonderful work on copilinidae, and she's right now examining the small size mollusca right now. Okay, and also we want to thank the tropical DC team. Okay, so that's a photo taken in 2014. 2014. Our chef, Jeff, Sarah, Philip Boucher, Xin is here, Helen Chen, and then my student, Janet, is here. And then, yeah, well, and so on. So thank you for your attention.